Yes, I am also on. Lukman.
Hello, everybody. Should we start? Do you want to say a few words or should we take it away?
Yeah, I think we can start.
Yeah,
Maybe Lukman will
my,
mute
my
other people.
Okay. Thank you to our hosts of the Computational Intelligence group for hosting us today to present a recent publication from our lab on predicting RNA structures utilizing attention from pre-trained language models. We are going to be presenting this webinar together with Yanis Papazoglou, who has been a student, a master's student in our lab, which was hosted together with National Kapodistrian University of Athens. And the name of the master's program was Computational Biology and Bioinformatics. We were very lucky to have Yanis do his diploma thesis in our lab. And this is the outcome of the thesis that was published in the Journal of Chemical Information and Modeling this year. So in the next slide, we will show you an outline of the talk. So first, we're going to introduce RNA structure, structures predictions with large language models in terms of predicting protein structure, predictions with large language models in terms of RNA structure prediction. We will be comparing with the state-of-the-art tools of what can be predicted in terms of RNA structure with current tools, and finally the conclusions and the future perspectives. And in the figure, you can see a teaser of today's presentation. First, we are going to show you that currently, just based on RNA sequence, it is very difficult to predict the structure of RNA with the current state-of-the-art prediction tools. However, as we all know, in terms of protein, taking into account the protein sequence and predicting the structure from the sequence, this can be achieved with using protein language models by utilizing attention. And it's very successful to predict the three-dimensional structure of proteins using AlphaFold and ESM fold. We know this already. And what we are going to be discussing today is in terms of having just the RNA sequence, can nucleic acid language models, can they be used to predict contact maps or structure? So in the next slide, we have the central dogma of molecular biology. Basically, the central dogma of molecular biology, what it tells us is that in principle, the principle that is that genetic information flows in one direction. It goes from DNA, transcribed through RNA, and then is translated into protein. So this process involves DNA replication, where DNA makes copies of itself. It's followed by transcription, where DNA's genetic code is copied into messenger RNA or mRNA, and finally translation, where the mRNA sequence is used to translate to certain amino acids that are then connected and exit the ribosome and become proteins. So RNA could be classified into coding mRNA, which transfers the genetic information. This is most as we know, mostly linear molecules, and then the non-coding RNA regulates then the gene expression and catalyzes protein synthesis, and these are structured molecules. So in the next slide, the non-coding RNA, which has this three-dimensional structure. So the goal of today's talk is to understand whether we can predict RNA structure. This has not been achieved so far. So in terms of RNA, in terms of structure, RNA has a primary structure that is consisted of nucleotide of nucleotides, the bases. So we know that we have the cytosine, uracil, adenosine, and guanine. These are the 4 bases that constitute RNA. And we have a primary sequence that you can see in an example in the bottom with the letters of the different bases. Then we have the secondary structure of RNA, where you can see that there are different patterns that are forming. There are hydrogen bonds between neighboring bases. There's a complementarity of bases. Not everything can form a hydrogen bond. So the adenine is bonding with uracil, then guanine is bonding with uracil, and then the guanine is also making hydrogen bonds with cytosine. And this can be expressed also in terms of contact maps. And we also have a dot bracket notation that is being used to represent the two-dimensional structure. And of course, as I mentioned, non-coding RNA has 3-dimensional structure as well. So tertiary structure, there's further interactions between distant bases. So in space, maybe in the primary structure, 2 bases are seemingly far apart. But in space, these bases may be coming together and forming interactions. And these could be visualized as contact maps and in atomic coordinates as 3-dimensional models. So we are exploring the notion that folding guidelines are encoded within base sequence. So we want to know if this is true in RNA. And of course, it is because we know that RNA has 3-dimensional structure. And we also know that 3-dimensional structure is bringing about the certain function that the molecule has. The next slide. So we are also exploring the current state of the art in RNA structure prediction. So we have different tools for the tertiary structure prediction and other tools for secondary structure prediction. So for tertiary structure prediction, different tools such as TR Rosetta RNA, the RAWfold, AlphaFold 3, DeepFold RNA, and FarFar2. Mostly AI-based methodologies, sometimes using also MSA, others not. Then we have secondary structure prediction tools such as Spot RNA2, RNA-fold, MXfold2, and RNA MSM. So we are going to explore whether these tools can be predictive, really predictive of the tertiary and secondary structure of RNA for unknown structures. Okay, in the next slide. So the question of today's webinar is whether by using natural language processing or language models, we can predict RNA structure. We know that for protein structure prediction, this has been very successful. Natural language processing, we all know it by now because somehow everybody became knowledgeable about what a language model is since ChatGPT came about. So we now know that there is a technique using artificial intelligence where you can actually use language models to create new texts and to come in dialogue with a person, and you can collect the information that is available in the literature or in the World Wide Web to synthesize new information. That is the subject of natural language processing. But it became apparent that natural language processing cannot only be used in our language, in English, for example, or in other languages, but it could perhaps also used in other types of languages. For example, protein sequence has 20 amino acids, and these are followed by specific letters. So these letters could be perceived as words that are forming different sentences. So proteins, in essence, could have their own language. So the question was whether natural language processing can be used to predict protein structure using these protein language models. So not only human language, but also protein language. So this came after AlphaFold was announced. So to overpass the limitation of limited structural data, Meta AI used a protein language model that was pre-trained on the abundant sequence data of proteins to uncover protein structure. And this was successful. A protein language model that is called ESM2 came about that learns biomolecular properties from evolution, including structural information encoded within the sequence. So we have a protein language that has a dictionary of 20 amino acids, and through the language model that was created by Meta AI, this could be used to gain structural insight, physicochemical properties, chemical relations, and this language model, ESM, through ESM Fold, then we could explore deep learning to recover these insights for precise three-dimensional structure prediction of proteins. Moreover, this natural language processing methodology was much faster than AlphaFold. ESM Fold was 6 times faster than AlphaFold because it didn't need to do the multiple sequence alignment, the MSA that was needed in AlphaFold. And finally, the result was of similar prediction accuracy. So in the next slide, we are now exploring whether similar pre-trained nucleic acid language models can be used to uncover structures. So not, of course, ESM, so Meta AI used ESM2 as a language model, and that there are in the literature other language models that are called nucleic acid models that are being available. And our question is it possible to create predictions of three-dimensional structures of RNA based on these nucleic acid models that are available in the literature? So at this point, I will give the microphone to Yanis to continue the presentation and the more technical part.
All right, thank you, Zoe, for the introduction and greetings to everyone. So I would like to start my speech with the main architecture of a language model and why they became such a success till today, actually. So they are based on a specific architecture model, which is called transformer, that was introduced back to 2017. So this transformer architecture, it was made out of two main components, an encoder part and then a decoder part. In the heart of this architecture lays an attention mechanism, which we are going to discuss in just a minute from now. Later on in 2018, another group actually designed the BERT architecture, which found actually a lot of success in biological language models that we are going to discuss also later. The only difference between BERT and transformer modules is that the BERT is only consisting from an encoder part, and it inputs the sequence and outputs just computationally interpretable but not human interpretable representations of this specific sequence. Now, as I told you before, the attention mechanism is the most important thing inside the architecture. They call it multi-head attention because it is happening in parallel in multiple heads and also in sequential repeats of layers. The main idea is that the sequence that we input inside the model, it's actually passed into three different linear projections, and we generate three different tensors, the query, the key, and the value. Two of those tensors, the query and the key, are used to generate a matrix, which matrix is actually their dot product. After this dot product generation, we pass it through a softmax to generate probabilities, and then multiply it again with a value matrix in order to create the final embeddings. Now, if we go back to this multiplication and the dot product between the two query and key values, we can actually represent it as a heat map or otherwise as a pairwise representation of the sequence towards its relations with itself. So that means that the model is actually learning what is the correlation of each amino acid of the sequence or nucleotide or base in our case, nucleotide base in our case, with itself and every other nucleotide of the same sequence. In other words, the model actually learns how to attend in each amino acid and how each amino acid is attending to itself and others in order to find meaning inside the language. As I told you, this is happening in parallel in a lot of heads and layers. Every attention mechanism, we can summarize it as a set of, let's say, attention centers. And researchers found that specifically in biological models, every one of those specific attention centers decodes a specific property of the language that the model is specific to. For example, in protein language models, some of those heads, they tend to showcase the structure. How do they do so? So structure can be represented, as I told you before, in contact maps. And actually, contact map is also a pair representation as well as the attention map. So some of those attention maps, they were actually mimicking so well the contact map, which was such an impressive event because those models literally they were trained only in sequences. They never saw structure, but they were able to decode it after understanding the language of proteins. So specifically in ESM, 19 out of the about 2,000 attention centers, they were revealing this structure of the protein. And when we read about that, we actually questioned this. Can the nucleic acid language models, in a similar way, decode the RNA structure without being trained in this? Because we don't have enough data to train them. So the first step we took it was to examine how many nucleic acid language models there are existing out there. So we found out 6 of them. All 6 of them, however, they had some, not all of them actually, 5 of them, they had some limitations. They were not even close enough to the parameters that the protein language models that you can see here, or the attention depth and the attention centers of those protein language models. So they were far more smaller, and also they were trained in far less sequences of RNA. So we had to test whether those can actually decode with these minimal. Yes,
Yes, I have a question there.
yes, of course.
It's about the concept of uh, nucleic acid language models, and then because we have protein language models, which I think most of us are familiar with, but then you bring in the new concept of nucleic acid language models, and then it looks like nucleic acid language models have not been like tested on RNAs, or they are probably not doing best. And as a computer scientist, I'm asking, what is the difference between these nucleic acid language models and the PLMs, but also RNA is not like nucleic acids. That's my question.
Okay, so first of all, the term nucleic acid language model is just a generalization. There are 2 types inside this general category, the DNA language models and the RNA language models. However, because the 2 languages are really similar, and most of the times we make the generalization that they have co-evolved, then we can classify them together, all right? Then for the second part of the question, the language of RNA or DNA similarly to the protein language, is having a vocabulary, actually not similarly, oppositely to the protein language, it has only 4 letters or 4 words inside its vocabulary. But the overall idea is exactly the same. We tokenize the sequence, we split the sequence into the monomers, which are the bases, or the amino acids for the proteins, okay, and then pass it over the same architecture from attention, and then extract either the attention maps or the output embeddings. Are you covered with this?
Sure.
Oh.
Perfect.
All right. So yeah, we had 6 of the RNA language models. And we found also one DNA language model, and we decided to try them out and see what is happening actually. What are they learning on? We directly found some problems. First of all, the RNA MSM. It is actually a new RNA language model, but it's not trained on sequences. It's trained on MSA. So it's taking a multiple sequence alignment instead of a single sequence, which actually drive us back to the main problem, which was inference speed. So if we try to test something with MSA, we lose the speed compartment of our theory. So we eliminated this from our tests. And then there was UniRNA. It was a really big RNA language model trained on billions of sequences, but its code never got launched, and the weights, they were never available. So we couldn't test it along the rest. So we ended up testing RNA-BERT, RNA, RNA-FM, RINALMO, and the DNA-BERT. So I told you before about RNA MSM. The RNA MSM, indeed, its creators tested if it's learning any form of secondary or tertiary structure from the RNA. And indeed, it's learning secondary structure. And when we passed over a sequence from the model, we actually managed to take an attention head that is actually similar to the contact map of to the target contact map. But what's happening with the other RNA LMs? So, of course, we cannot just take random sequences and pass them over the language models and then expect to find the similarities. So we needed to find a way to make the whole analysis quantitative. So we found this really nice formula, mathematical formula that they suggested in a preprint in 2021, that's correlating exactly that, the attention maps with the contact maps in the base pair level. So what this formula actually tells us is that for each base pair I and J, we are taking all its attentions under a specific threshold, and then we divide it by the attention heads, the overall attention heads of the language model. In other terms, to simplify it, we take all the attention that is actually giving us structure, divided by the all general attention of the model. So that was one way to estimate how much structural information is inside the language models. And then because this formula is actually directly giving us the alignment, it doesn't more sophisticated searches inside the attention maps, we decided to do some simple machine learning classification tasks in order for the classifiers to go inside the attention weights and decide if other hidden patterns to our eyes can actually be used to show as the structure.
Sorry, on the previous slide, on that mathematical expression.
Yes.
So when you're quantifying attention, when you look at that summation where we have f of I and j, I'm assuming like you have these two sort of like elements within the sequence I and j, and my first impression is, does that represent co-evolution? But also another thing, when I see the angle theta, I'm thinking about like, are we now talking about like 3D structures whereby we are looking at the angles between given sort of like elements?
Yeah, so in this level, we are only trying to find structure under contact maps. So into 2D level, not any 3D, no any angles or something similar to that. So things are really simpler. So what we need to do, we have two kind of matrices. Okay, one it's the contact map, and it's a binary matrix, 1 and 0, with pairs I and J. And then we have the attention map with the same pairs I and J. And those attention maps are actually probabilities, 0 to 1, okay? So we take for every pair from the contact map and the attention map, we take this function f, which is either 1 or 0, if it's structure or not, multiplied by the attention, the possibility that the attendance of the module. If it's 0, then it's going to eliminate it totally from the calculation. If it's 1, it's going to be calculated, okay? This is happening all over the J's and the Y's of every sequence, and the I's, sorry, of the sequence, and all over a dataset X, okay? And this all over the dataset, the I's and J's of the attentions, we get a probability of how many attention weights are actually showing structure versus all the number of attention.
Okay.
If it's
So at the end
clear.
of the day,
Hmm?
okay. So at the end of the day, it is a probability.
Yeah, it is a probability. It's P of F in the end. Yes, it is a
Okay.
probability. Yeah, because
My confusion is about the angle, like theta. Okay, not I don't want even to say I'm biased. Like when it comes to angle.
theta is not an angle here. Theta is a threshold.
Okay.
So this threshold is actually telling us how many attention weights to get inside the calculation or not. Usually they set this theta to 0.3. That means that the less important attention weights, because they are noise actually from the language model, they get eliminated as zeros. Okay, so we only measure the larger attention weights versus all the attention weights. This is theta. This is just a threshold. Yeah.
Okay. Perfect.
Yeah.
Thank you.
All right. So, as you said as well, this is a probability. So this is just an alignment to see how much attention it's actually showing us structure. So we needed to find something more sophisticated, something that dives into the attention map and seeks for patterns and brings us back structure. So we did a classification. We converted to a classification task. So in this case, we took every I and J as a sample, and as features, we used the attention weights that was assigned to that pair from the language model, and then we made the classification algorithm to decide if it's a structure or not. In order to do both, either the statistical analysis or the machine learning, we needed a data set, actually at least two data sets. One, it was for the tertiary structure, so we needed to go to the protein data bank and the nucleic acid knowledge base. We curated every possible RNA model inside there. Then with Python, we are actually cleared the multiple chains, kept the single models. If it was NMR, we kept only the first model, and then we deleted all the invalids. So there's a problem with language models. They have some criteria for a sequence to be inputted inside. It needs to have only the 4 bases, not any modified bases or any unusual bases. So any sequence with those needed to get out of the dataset. And then we needed also to discard any structures that they had gaps. They had missing residues because those were messing up with the contact map and the dimensions of the target and the input. In the end, we did clustering analysis with the algorithm CD-HIT. We used a similarity score of 80% to cluster the similar sequences inside groups, and then we kept one representative from each group. We did sequence similarity only and not structure similarity because language models are just aware of sequences, not structures. So that was the only needed clustering here. In the end, we ended up with 425 tertiary structures, which we can see it's not even close to enough the data that someone needs to train any model. But anyways, we continued to secondary structure. We tried to curate again the structures, the secondary structures of RNA to see what's available. Here things were more positive because there are 3 open source secondary structure datasets. They are called RNA Star Align, Archive2, and BPRNA. So those, they contain about 150,000 secondary structures. problem here it is that they are computationally estimated along with some experimental work. They are not 100% resolved as the PDB database, though a lot of other works used them in order to train AI models, and it was working, so we decided to go with them as well. Again, we cleaned them from invalid bases, and then we did clustering, sequence clustering with the same similarity. We ended up with 30,000 secondary structures, which was better than the tertiary dataset. Now we had to create the target, which was the contact map. So for the contact map, we had two types of contact maps, the ones showcasing the 3D structure and the one showcasing 2D structure. For the 3D maps, it was just a measurement of all the distances, the atomic distances between each residue pair, and then kept the closest one. That gave us the distance map of the molecule, and then by setting a threshold, we converted to a binary matrix contact map. And then for the 2D maps, we took the dot bracket secondary structure given by the dataset, and we just with code converted to a binary matrix as well. So to that end, we had the targets ready as well. We needed to do just one final touch that Meta AI suggested. So we needed first to transform the contact maps. We needed to delete the diagonal line. So this diagonal line is the direct neighbors of its residue or amino acid. That means the primary structure of the molecule. And because this is known to the language model, because it's inputting the sequence, we needed to take it out because otherwise it would give a lot of noise to the model. So we deleted the diagonal. Specifically, we deleted the first 4 neighbors of each residue. And then the second transformation we needed to do, it was for DNA-BERT. So DNA-BERT, the DNA language model that we chose to check, it had a minor difference than the RNA language models. It is splitting the sequence into 3 mers, okay, 3 bases together. So we needed to find a way to convert the contact maps from 1 mer contact maps to 3 mer. So we used the kernel window to screen all over the contact map, and every time that it was looking at a binary target 1, it was printing 9 of them around in order to create around the window in order to create the 3 mer contact map. That being said, we had to generate the features now and the samples for the machine learning training dataset. So we passed over the sequences over the language models. We took the attention maps. For each pair, I and J again, that it was a sample, we took all the attention weights, and then from the contact map that we just created, we took the label either 0 or 1. And that was the dataset generation, which had a major problem because of the nature of RNA, actually. So we had a huge imbalance problem.
Yeah, so some more questions here about the previous 2 slides, I think. Let's go back to the previous slide.
Okay, this one.
Yes, that one.
Okay.
About the generation of the contact map, and one wonders, because we have the sequence and where we are like sort of mapping corresponding bases or nucleotide, like when you have on the vertical and on the horizontal, and then you sort of like encode where you have 0 and 1. And one is asking, we have this sequence of the RNA, and we are creating a contact map, and we are creating 1 and 0. Which information is coming out of this contact map? Is it like, because I'm assuming we are having a sequence sort of like 2D, like which information is the contact map giving out? Like, is it like how close, like
Yes,
Each,
exactly. It is actually which of the bases are interacting into 3D space. So
oh,
whatever is a binary, it's a number one label here. That means that those two residues actually they are doing hydrogen bonds together and they create the secondary and later the tertiary structure of the molecule. So when you have a contact map, it's actually showing you which bases are interacting directly into 3D space. It's just a simplified representation of the structure, really economical and simplified representation of the structure instead of having 3D coordinates, for example.
okay. So,
And we are using them, of course, because they have the same dimension with the attention maps that the language models are outputting. So it was just, we didn't have any other option. So we just had to go with this pairwise representation.
okay,
Yeah.
so the attention, the contact map is generated as a prediction from the model, or it is part of the input within the
So
model.
right now in this step, we just generate it from the dataset, from the data that we have available. But later on, we try to use the attention in order to predict this attention map. Yes, it is the
Oh,
target eventually. Yeah. So for unknown molecules, we try to predict exactly that, the attention map, which is later can be translated to structure for researchers.
perfect.
Yep. So, yeah, having the dataset, the problem was that it was now imbalanced. Specifically, we had like 95% of class 0, which means not residues that they are not interacting in 3D space, which is totally normal for the RNA. So we had to find a solution for that. Really straightforward. We did some randomized undersampling here. So we decreased the number of the samples that had class 0 to be equal with the number of instances that had class 1. We gathered some millions of samples like that out of the initial sequences, and then we had to decide which classifiers we are about to use. First of all, we use the logistic regression, which is the one that Meta AI also was suggesting. And then we decided to go with tree-based classifiers as well. We tested decision trees, random forest, and the XGBoost, which allows for GPU parallelization. And then we did two neural networks. Okay, the first one, it was the classical multi-layer perceptron, the MLP. But the other one was a convolutional neural network, which we are going to discuss later on. In the end, how did we evaluate the prediction? So the target, the thing that we are predicting, it's a binary contact map. Okay, so if we flatten it into 1D, then we can take every sample and compare it to the true structure. And thus we can calculate the recall and precision scores and the F1 score, which was the score that we mainly used for our evaluations. Later on, we also used the MCC, the Matthews correlation coefficient, because it seemed to be a little more better actually to handle imbalanced datasets. Starting with the results, first of all, okay, we had the techniques, but we didn't know if they're working. So we decided to go back to the protein language models and test them first there to see if we indeed can do this. So we tested all the versions of the ESM fold to begin with, from the smallest one to the second largest one. And this is actually the results of the mathematical estimation that I described before. Those are actually the attention centers of each version of the language model. You can see here it's the smallest language, and then it goes on as the parameters grow. In every one of them, even in the smallest one, there are darker spots, which means that in those centers, in those attention centers, the probability of the attention to be aligning with structure, it's really high. Those darker centers, they are actually touching even 90%. So that means that 90% of the pair representation, it's actually indicating for structure. We did the same for the T5, the Prot-T5 language models, two versions of this, one trained in 45 million protein sequences, the other one in 2.1 billion protein sequences. Both of them also showcased a lot of attention centers that they are actually showcasing for structure. In both cases, it was only the attention centers of the higher layers that they were revealing most of the structural information, and both of the ones designed either the ESM or the Prot-T5 language models, they commented on that because structure is a really complex biological and biomolecular property. It is actually needed a lot of repeats of the attention mechanism until it is able to decode it throughout its attention. Then we did the machine learning, the logistic regression to those attention weights for the protein language models, and we used only 20 proteins to train the logistic regression classifier. And you can see here that from the target contact map, only trained on 20 proteins, we managed to get really good representations close to the original one of this structure. The F1 score here, it was ranging around 0.8 or 80%. These scores here are actually from a logistic regression model that was trained in 20 proteins, and then it was tested on 15,000 proteins. So it was impressive that those models actually decode the structure of the protein.
Ionis,
Uh-huh.
the previous slide, when you look at that panel with the contact maps, I'm seeing like the arrangement is sort of like uniformly diagonal, and one would wonder what information can be obtained out of that. Like we have that sort of perfect alignment along the diagonal, and does that mean anything?
Yes, so this is really important information actually, and this is the exact information that AlphaFold is using from the MSA. It's decoding for the MSA in order to predict the eventual 3D coordinates of the protein. So what Meta AI did here, they totally discarded the MSA, which is really slow. They used those representations here, the predicted contact maps, in order to use them as the pair representation inside the EVO former and later to the IPA, the invariant point attention module of the AlphaFold, to predict the 3D coordinates. This is why actually MetaAI ESM fold is 6 times faster than AlphaFold 2, because they managed to obtain those initial representations of structure that they later encode more and bring as 3D coordinates. So this is actually the novelty. This contact map here, it might not showing a lot of information compared to a 3D structure of a protein, but for a model, this mathematical representation here, it's super informative in order to use and predict the 3D coordinates later.
All right.
Yep. And and working a little bit further with that, I managed to see that indeed this representation here, it's the most important thing in the AlphaFold architecture. Like without this, you can never have the success that AlphaFold had.
Wow.
Then we did something as a validation. We took the statistical estimations that we did, and also the coefficients of the logistic regression, and show if they match, if they find structural information in the similar layers and heads. And indeed, both of them agreed that the later layers of the architecture were the ones that giving the most of information. Proceeding now to the RNA, we can start first with the tertiary structure of the RNA. So we use those 5 nucleic acid language models. We did the same calculation. We get some results, but some of them, they were really strange. Like this one here in RNA-FM, it was giving 100% possibility. That means that this head theoretically has the attention, the contact map resolved like 100% there. With further estimation, we managed to find that this is an artifact actually happening by the attention mechanism. So when the attention is something similar to that, and we said those darker points here are actually probabilities. When those probabilities are really, really high, like 1 or 0.99, when we use this threshold to eliminate all the background noise, we end up with this specific signal, which if it is inside a contact pair, then it gives us this 100%. So in order, which is an artifact as I said before, so in order to solve this problem, we totally eliminated the theta threshold here for the RNA and DNA language models. And this is happening because apparently, as you can see in the plots here, there is not enough structural information decoded inside attention. So we need those lower attention signals in order to find something meaningful inside the model. So while we were eliminating those, we were just throwing away any possible source of information. Anyways, with these not so positive results about tertiary structure, we tried to print some of those attention maps from the biggest of the RNA language models, RNA-FM and RINALMO. And as you can see here, there was no evident alignment, like it was something that weren't similar at all. When we did
Hence,
machine learning, yes?
on the previous slide, where we have the many contact maps, and we on the previous slide, the previous one, yes, I think this one. When you look at the different models and the kind of contact maps that they put out for the RNA, it looks like the prediction for the structure is really, really bad. Like even when you increase the layers or you go for the higher
Exactly.
layers.
Exactly.
And like my take, like this is happening for RNA only, and why? Like, is it only happening for RNAs? Like, you cannot predict like the tertiary structures using these models. Is that what it means?
Not 100% like that, but it means that the language of RNA is actually more difficult to decode. And once we have these kind of architectures that they are not even half the size of the protein language model architectures, it means actually that the current models, the language models for RNA, they are not capable of finding structure inside their attention. This is exactly the outcome, at least tertiary structure, okay? Not to generalize a lot, but yeah, this is exactly the meaning. And that was proved also when we did the machine learning. So we trained the some classifiers with the language model attentions, first on 20 structures only, and then on all the 300 um, tertiary structures that we collected, and the outcome was random. It was not it was not anything meaningful. That means indeed that the language models weren't enough to learn any structural information about the tertiary structure specific inside their attention. And then we continued with secondary structure because secondary structure it's considered as a simplified version of the RNA structure. So we said maybe they are able to find secondary structure but not tertiary. Again, the smallest ones like the RNA, RNA BERT, and DNA BERT weren't including any information inside. The RNA ephemeral in Almo had some centers, attention centers, and when we printed the maps again, we found out that they indeed have some alignment. By the way, I notice now this is a wrong figure. Please visit the paper. It's there's a figure there. So this is the tertiary structure again, but it should have been the secondary structure. The outcome of this is that the attention maps, they are actually kind of aligning with secondary structure, but yet we do have a lot of noise. When we trained the machine learning classifiers for the secondary structure this time, first for 20 structures and then for the whole dataset of 25,000 molecules, we ended up again with close to random predictions. Sometimes we did have some outliers climbing the F1 score, but still those were just some extra instances. So again, the maps, when we printed them, they were clearly random. The only thing that we can find out from here, it was as we increase the architecture size from DNA bird that's the smallest towards the renal one, which is the biggest, we can see a clearance in the patterns, which means it's a good sign that actually the models, the bigger they are, the more capable they are to decode more properties from the language. We did also something else in secondary structure predictions. We split the training dataset of 25 into batches, first of 20, then 2,500, and goes on to the whole dataset. And then we trained and evaluated the predictions in order to see if there are any fused capabilities of those RNA language models. Here, the outcome was that they might have, but most probably they don't, at least at the current level that they are not capable of finding structural information. Continuing on with those results, we decided to go a little bit further and build something more advanced. So we decided to go with a CNN, convolutional neural network. So instead of taking every I and J pair and then decide if it's a binary contact or not, then we decided to take the whole attention map altogether, pad it to a specific fixed dimension, and use it as an image. So like an image classification task, we process them through convolutional neural network with couple of convolutions and pooling layers, then to a fully connected layer, and then initially we transformed the 1D vector to a contact map, trained it to the full dataset of 25,000 secondary structures, but still it seemed that the model, the CNN, was not capable enough to find patterns indicating structure inside those models. At that moment, Rawfold, a publication in Nature, came out. What they did exactly the same with one of these language models, the RNA FM, okay? But what they did, instead of any convolutional neural network or any machine learning classifier, they just stick the architecture of AlphaFold 2 inside after the language model. So as you asked before, they took those pair representations from the attention maps from the language model, and they used them to actually make the model make anything informative for structure. They indeed managed to succeed with a lot of limitations still, like they cannot predict non-synthetic. They can only predict natural sequences, not synthetic sequences. So they miss a lot of accuracy there because the language model is trained only on natural sequences. And then another problem is that as long as the sequence length is increasing, they lose accuracy as well. But it was a nice idea that if you add a really, really intense architecture after the language model, maybe you can get some predictions. We left it there. We just did an eventual benchmark. So we predicted some structures in an independent test set of 33 structures from the PDB, excluded from the training dataset, of course. And we predicted their structures with TR Rosetta RNA, Rawfold, AlphaFold 3, the FARFAR algorithm, or DeepFold RNA. Those 3D predictions, we converted them to contact maps and compared them to the language models plus the random forest classifier that we trained earlier on. And of course, it was expected. We saw that they cannot reach the accuracy of the other molecules. We did the same for secondary structure, state-of-the-art methods like spot RNA, RNA fold, MX fold 2, or RNA MSM. Here again, the same results, only there was one thing. The other language model that is actually inputting MSAs, it had actually really similar accuracy, F1 score actually, with the Rinalmo language model with the random forest classifier attached to that. With all those being said, I would like to give you this table again. You can see the clear difference that the parameters that the protein language models they have and the length of the attention mechanism and how small the current nucleic acid language models are. So we decided that it was the time to take action and to bring some novelty here by trying to train a new model, a new language model trained in as many RNA sequences there are existing out there, and by increasing the architecture to something closer to the protein language model by actually training a model with billion parameters. The results for that are about to come out. But the results for this work that I presented till now, it's indeed that the protein language models, they do find structure in an unsupervising way from just trained on sequences. And then they present also really strong few-shot capabilities, which means that if we fine-tune them into like 20 proteins, we can get really refined contact maps out of that. The RNA LMs for tertiary structure, they really contain no information at all at this level, except if we increase the architecture, of course. For secondary structure, there is a little bit of development in the predictions, but still we are not there. The main limitations we found out, it was, first of all, that the architecture was small. The data used to train the language models, it was at least 7 times fewer than the ones used for the protein language models. And then it was the nature of the language. So the RNA language is only consisting of 4 bases, which means 4 words. So it's really, really difficult to decode the language with only 4 words. While when you have the protein language, for example, which consists of 20 words, 20 amino acids, it's kind of easier, or the human language, which consists of millions of words. And in the end, the RNA nature, so RNA molecules and their structure is really, really flexible, and most of the times it gets stabilized by interactions in their quaternary structure, which means that other molecules come and stabilize their structure. So it might be that a single language model trained on sequences, it is really difficult for a small one at least to decode this level of information. The outlook, as I said before, we really need bigger molecules, billion parameter molecules actually, that can bridge those limitations that I just explained and bring something really accurate to life. In this time, I would like to thank everyone contributed to this work and consulting, Dr. Cournia as well, the postdoc of the lab, Alexis Chatzigoulas, our collaborator, Yorgos Tsekenis, Vasilis Liakopoulos, and Evi Geka also for their consulting. And thank you very much actually for your attention.
Thank you. Also, I would also like to thank the Amazon Web Services for providing computational time, and GRNet also providing computational time together with Amazon Web Services and our funding, and of course the members of our lab for general support, and you for hosting us.
Pleasure is ours. I had a question, but let us first go to Savia. Savia, your hand is up. Please unmute yourself and put your question forward.
Thank you so much. for this kind of opportunity. It's my first time attending this
Oh, we can't really hear you.
The vibration. Come again, Xavier?
Yes, I was saying it's my first time attending this.
Okay.
And I reckon that there could be a mailing list or something like that to inform people through. I would like to have a prior access to the papers that will be presented in the subsequent meetings, and I would love to be an active member of this.
Okay.
Yes.
Okay, Xavier.
I don't know what protocol. I don't know what protocols I would follow in order to
Okay, Xavier,
be a full member.
I'll answer that straight away. We have a Slack community where people like receive updates too. We have a website. I'll share the link in the chat. Yeah, but we have a Slack channel where workspace, sorry, where everything is there, and we have a YouTube channel also where previous recordings have been uploaded. This one, because Professor Zoe asked to record and put it on their channel, so there's no point for us to have it. But we
But
will have.
I can also, you know what I could do? I could send you the recording and you could also upload it in your own channel. That's not a problem. I'll send it to you when we finish.
Okay. Okay, thank you. Thank you very much. Yeah, so Xavier, I'll put the link in the chat.
All right.
Yes.
There is a question in the chat actually from Herbert.
Oh, Herbert, I think Ionis,
That question was sometime 30 minutes back. I don't know, maybe he really try and answer.
Yeah,
But I
Yanni,
also wanted to ask that you showed a significant improvement in the performance over the existing methods on your benchmark, but your limitation slide mentioned architectural constraints and the scarcity of high quality data. So in the absence of the large, diverse data set of experimental and verified structures, did you explore any alternative strategies such as transfer learning from other modalities or incorporating explicit biological priors into the model's architecture to help it generalize to a new unrepresented RNA families?
So this is a transfer learning approach actually. So the language models are only trained on sequences. They never see structure. They are never constrained or biased by any structural data. The only input that they are actually receiving for their training is the input sequence, just like that. And then they are capable enough to decode the structure in an unsupervising way by themselves. So, yep, that was our initial task, to find a way, like a transfer learning way, to actually eliminate the issue of data scarcity and actually be able to predict structures without the need to train the model explicitly to structure. But as we found out, the current means that we have, the language models available, they are not capable because of their architecture. There are architectural constraints.
Okay. Yes, Ionis, my question was about the choice you made somewhere between the MLP and the CNN, I think for secondary structure prediction.
Yes.
Yes. So I'm wondering, CNN, we are used to a CNN in terms of visual sort of analysis, for images. And it wouldn't be my first choice for this prediction. So why the CNN, and how is its performance in that regard?
Yes, performance was not good. You can see it from here. But the idea and why we used the CNN, it was when you have an attention map, it's, as I told you before, a pairwise representation. So if we take every attention weight as a pixel, as they do in image classification, and we put as channels the different layers of the attention map, that means that we have multiple channels based on the attention mechanism that we are using. And then the actual resolution of the image or attention map, it's actually the maximum sequence length. That was just an approach that we were like, okay, we have something that we can visualize as a map, the attention, and then something else that we can also visualize as a map, the contacts, and then we need to come from one side to the other. And because they look like, at least to our eyes, as images, why not trying the image classification? So we ended up thinking like that. Maybe if we represent as pixels, every pair as pixel, maybe it would have give something informative. But in the end, yeah, it wasn't enough. I feel like that it was, if it was for another architecture really, but really heavier, it might have found something to decode. But in this case, no, it wasn't enough to get some outcomes still.
Okay, I really say I think it's an intuitive way of thinking about it. Unfortunately, it did give great results, but I think it is quite ingenious. Um, I think that's all from me.
I
And
should also say that our code is available. I mean, if you download the paper, the code is there and it's free. Whatever
okay,
you saw in this presentation, the code is uploaded in GitHub, right, Yanni?
Yes, it is on GitHub.
And if you can't find it or if you want extra information, please contact us. All the code is open access and open source.
definitely. I would implore anyone to look at the code, like look at how these networks are retained, the attention maps, and the manipulation going in there. When you look at that code, it's really an exciting way and learning more. So I implore members to always look at the code, and definitely I'll check it out by all means. My attention will be on the attention maps and the contact maps.
Mhm. Mhm.
So
Yes.
thank you, Ionis and Professor Zoe, for really saving up time, and plus the lab members who have made it here, and to our members, most of them have left. And thank you very much, and thank you very much indeed. Can't thank you enough.
Thank you very much. And we'll be in contact for sharing the presentation, the recording, and the slide.
Thank you very much. That will be very, very, very appreciated.
Thank you. Bye, everyone.
Thank
Bye-bye.
you.